Hi, welcome to clickmyproject.com. This video will lead us to the demo of a combination of feature extraction methods with an ensemble of different classifiers for protein structural class prediction problem. Important information about overall folding type is revealed by structural class of the protein. Pattern recognition based approaches are used for many of the enhancements. Overlapped segmented distribution and autocorrelation based feature extraction methods are used to provide discrimination information of global and local. Important 15 attributes are selected from the physicochemical based attributes. Different classifiers are used to show the accuracy of protein structural class prediction.
The amino acid sequence are the input for this project. The group of amino acid sequence are combined as proteins. These proteins are ordered in a PSSM, that is the position specific scoring matrix. In the position specific scoring matrix, there is a method called sequence based method. This is the feature extraction method. In this sequence based feature extraction method, there are two types. They are PSSM AC and semi AC. These two types are used as a feature extraction methods, and from the PSSM matrix we can get the data like consensus sequence. These consensus sequence are retrieved from the PSSM, that is the position specific scoring matrix. From the content of consensus sequence, the physicochemical based method is retrieved. In the physicochemical based method, there are two types. They are segmented distribution and segmented autocovariance. And then the FCBF, that is the fast correlation based filter. This is also a feature extraction method. Totally there are 5 feature extraction methods are combined to form the combined feature vector. In this combined feature vector is the input for the classifier and the clustering. There are ensemble of classifiers are used in this. They are ADA-boost, MLP, that is the multi-layer perceptron, naive Bayes, logit boost, SVM, support vector machine, linear regression, and a priori. These 7 classifiers are used to classify the proteins present in the dataset. And then for clustering the proteins in the dataset, the k-means clustering is used. Let us see the demo of combination of feature extraction method project. Here we have to load the dataset. For that, click browse button and select the dataset. This is the combination of amino acid sequence and form the proteins. Then press next. Here you have to click load database. Now only the position specific scoring matrix is created and stored in the database. Now click OK and then press PSSM data. Now you can see the data that is presented as a matrix format. Now press next and calculate the PSSM AAC. This type of method is present in the evolutionary based composition feature group, and then press next. Here we can find out the PSSM AC. It is present in the evolutionary based autocovariance feature group. These are the equations. These equations are used for calculations. Then press next. And then the consensus sequence. These consensus sequence are get from the original PSSM. The argument max, that is the maximum value, is find out and it is replaced with the minimum value. Now the minimum values are replaced and the maximum values are presented. This is the consensus sequence extraction method. And press next. Now we can find out the global density. It is present in the overlap segmented distribution approach. Now press next. Here we have to find out using the 15 features forward direction, and then press next. The 15 features from backward direction is also present in the overlapped segmented distribution approach. Now press next. And then the second method, it is the 7 features from forward direction. Then press next. Then in the overlapped segmented autocorrelation, the 7 features from the backward direction is also calculated. Both these 15 features and the 7 features are used to find out the general composition. Then press next. Now we can find out the h of x that is present in the FCBF, that is fast correlation based filter method. Find out the h of y value. Then press next. And then here we have to combine h of x and h of y, that is h of xy. Using that, IgXY is calculated. It is the entropy of xy. Then press next. Ig means information gain. Now we have to find out the symmetrical uncertainty. In that symmetrical uncertainty, we have to find out the threshold. If the amino acid is below the threshold means then it is not present. If the threshold is above means then, if the amino acid value is above the threshold means then it is present. Then press next. Then find out the descending S list. It is used to find the S best features from the fast correlation based filter algorithm. Then we have to classify. In classification, we have to see the combination of feature vector, that is the CFV. It have finally 10 features. Our input contains 20 features. We have to reduce it into 10 features and then press classification. In classification, the first one is Addons classification. Using that Addons classification, we have to find out the threshold and then press next. And then in the logic boost also we have to find the threshold, then press next. In naive Bayes we have to find out the C value first and then the Px by C value, then press next. Using that P of x and Pc, we have to calculate the naive Bayes classification method. Here we calculate the threshold value for that and then press next. And then the multi-layer perceptron. This classification also contains the threshold value. Then press next. In support vector machine, we find out the threshold value for all the proteins. Then press next. And then in the classification algorithm, a priori algorithm is used. In that, the candidate item set 1 is calculated and threshold is found out for that item 1. Using that, we find out the frequent item 1. After that, press next. From the frequent item 1, we calculate the candidate item 2. In that, we find out the threshold value. Using that, we find the frequent item 2. Then press next. Then the linear regression method is used. In that, we find out the n value and summation of x, summation of y, summation of x into x, summation of x into y, and then we have to find out the slope value for that and find out the intercept and regression. Using that, we can draw the graph and then press next. And then we enter into a classification method. For that, press classification. In that, this is the overall classification. We get the proteins that are classified at last and then press next. Now we take the k-means algorithm for clustering. For that, we have to create the 4 clusters. Then press calculate. Now the proteins are separated into 4 clusters according to the similarity. Then press graph. It is used to see number of proteins in each cluster. Thanks for watching this video.